Hey, good morning. Please take a seat. Let's start. Okay. Okay, hope you are okay after the dinner yesterday in the nice place. Hope you enjoyed. Sorry for the weather. It's not my fault. It's too hot. Humidity is bad here in Barcelona, but don't worry. Okay, so thank you for being here. Today is the second day, and we will start with James Sharpe, who is the coordinator of the European Biology Molecular Laboratory here in Barcelona. His lab is a multidisciplinary lab, having physicists, mathematicians, statisticians, and computer scientists. And his work is mainly devoted to study and compute or create computer methods and simulations to study the limbs in mammalians. But I guess that today he's going to present a different, or is related more or less, but using techniques, spatial transcriptomics, which is interesting for all of us. And the idea is that to see what he's developing, the new techniques and the new devices that they are generating to obtain this data, and maybe in the future, pues, any one of us can implement a package in Bioconductor to analyze the data that his lab is generating. So please, thank you, and the floor is yours. Thank you.
Great. So thanks very much for inviting me. It's a pleasure to be here. And indeed, I'm going to, just make sure this is working. I'm going to talk about a kind of spatial transcriptomics, or a sort of a spatial omics technique that can be used for transcriptomics. And in fact, it's something that we've developed. And what I'm going to do for the first sort of 5 minutes or so is give you background about our lab and what we're trying to do, to put it into context. And then I'll go on to this technique, and then I'll answer any questions. So we're interested in cells, but in fact in multicellular situations, so tissues. And a tissue is not just, a collection of cells. A tissue is something that is interesting from a particular point of view, that it is spatially organized. Now this is a limb bud. This is the structure that we study. So when your arm was, when you were an embryo of about 6 weeks old, very young actually, this is what your arms and legs each looked like. And it's quite a lot of cells. So it's about at least 100,000 cells at this point. So we don't actually care that much about every single individual cell. What we care about more is that, for example, in this image here, the cells all look more or less the same. It's just when you reveal the expression of certain genes, like the green gene here, this is our favorite gene, this is SOX9, you can see that it has a specific pattern. If I go back, again, if you looked at almost any other gene in the genome, you will not see anything more than sort of this. This is the point of pattern formation, decision making, when certain cells have to do one thing, certain cells a different thing, and in fact it's only a few genes that initiate this. Later on there are cascades of thousands of genes, but we're interested in the moment when the decision is made, and the decision does not involve thousands of genes. So this is what SOX9 looks like at a certain time point, and you may not be able to guess from this what structure it will form, but of course we're also interested in time. So a little bit later, this green gene, SOX9, looks like this, about a day later. And you might be starting to guess what part of the arm or leg it's going to form. But if you look at it 1 day later, then you can see what it's starting to form. So this gene is switched on in the cells that will form your skeleton. 1 bone here, 2 bones here, 5 sort of fingers here. And as I say, if you don't look at the right genes, you won't see anything, right? So the genome as a whole is not going to tell you that much. But later on, thousands of genes end up being regulated in this way. So our problem, to put it into context before I go into the spatial transcriptomics, is at this kind of scale. Right? We want to understand at a tissue level which genes are on and off where. And of course, it's not just on or off, it's levels. So it's high here and it's maybe off here, but there's multiple different levels of gene expression around here. Now, how limb development works, or any organ forms, is actually clearly a multi-scale problem that involves genes, it involves regulatory networks, gene regulatory networks at the molecular scale, but it also involves cells and cell behaviors and cell activities and what they do. For example, how fast they divide or in which directions they move. If cells were not making careful decisions about how to move, you wouldn't end up with a specific structure like an arm, you would end up just with a blob. And then at the higher level, the sort of macroscopic level of the organ is also relevant. Of course, this is sort of, in one sense, the result of all of this process, but actually it feeds back onto the molecular thing. And something that I think is hugely important in biology in general, it's very obviously important in development, is that molecular events are actually controlled by macroscopic events. I mean, the simplest way to think of that is something like size regulation. When you're developing a heart, the heart, for example, has to grow to a certain size. Now, the genes in each cell need to know whether to stimulate proliferation or not, but a single cell does not really have direct access to know how big the heart is. The heart is orders of magnitude bigger. Somehow there's some communication from a large scale, macroscopic scale, down to the molecular scale. And this is what makes biology interesting and fun, that it's not like a hierarchy of causality going from molecules or genes up to larger biological processes. It's a total feedback from small scale, medium scale, large scale, and back again. So this has been recognized for a very long time, at least 73 years. This was written by Alan Turing in a paper when he actually wrote a paper on developmental biology, which many people don't realize because he's so famous for so many other things. Minor things like inventing the concept of the algorithm and contributing to the development of the computer. He did a paper on embryology as well. I think largely because he was obsessed with what is a thinking machine. Of course, he's famous for the Turing test and artificial intelligence. And it seems he sort of came to the realization that, well, there is one thinking machine that we know of in the universe, and that's the human brain. How is that built? And realized that it's embryology, and started thinking, oh, how does embryology work? How do you build a thinking machine? Because he wanted to build an electronic one. Anyway, he has written this paper, and it's absolutely fascinating, very insightful. And he pointed out that there are both sort of chemical processes and mechanical processes necessary, but for the moment, back in the 1950s, we'll just sort of separate them to keep things simple. And in some respects, the field has spent the last 7 decades still trying to get these two things back together again, the mechanical and the chemical. Many groups are working on that now, and this is sort of one of the motifs for my lab. So we work on limb development, and you can consider a whole range of processes at the chemical level and a whole range of processes at the mechanical. And we end up having to spend a lot of our time just describing or quantifying what's going on. So this is a sort of roadmap of our lab, as it were. On the chemical side, we've got observations or raw data, and that's ultimately what I'm going to talk about more in this talk. So gene expression patterns, for example, and other things. From raw data, you want to turn it into sort of interpretable data, into databases and atlases and mapping. This does not necessarily explain something, but it helps to get there. And the same then on the mechanical side. We have to study cells, we have to analyze what they're doing, we have to, well, here is at the molecular side, so we're trying to develop methods for smoothing together data about this gene dynamics over space and time. And on the mechanical side, we have to quantitatively measure what is the shape change accurately, and we've developed new imaging techniques for that. And try also to work out what are the internal tissue movements. And so we use clonal analysis here. Here you can see little collections of cells that have been labeled. Each little group of cells are the cells that came from one single original labeled cell. And then we have developing, as was mentioned in the introduction, lots of new computational tools, basically all in-house developed software to analyze this. Now, all of this is just sort of collecting data, and we're sometimes proud, right? I mean, sometimes you hear from microscopy things of how many terabytes or petabytes or whatever we collect. In reality, we don't want terabytes and petabytes of data. We have to generate it to sort of see what's going on, but I would propose, and in fact, we almost started discussing this just before the talk now, you know, data is not what we're after, data is what we need. Because what we're after is understanding. And understanding, for me, comes from mathematical models, essentially. So here we are really putting a lot of effort on building mathematical models of the gene regulatory networks and developing simulations. We're also developing simulations of the mechanics and cell behaviors and how they integrate to create this sort of complete whole. So I do think that really, as a sort of side comment, as you sort of go down and titrate and sort of reduce and concentrate what you really want out of all of these terabytes of data, the final understanding is something you can put on a USB stick for sure. In fact, these computer models are small and they are differential equation based, and you have parameters, initial conditions, and if that doesn't work, then you basically don't understand your system, I would say. So I'm going to talk about this thing called C3PO, but let me just first give you a sort of a glimpse into, well, the future for us, which is what we're really trying to work on, which is these agent-based multi-scale models that will integrate all the way from the molecular, cellular, and the tissue level. This is a simulation that actually runs, so we've written this software called Yala, which sort of natively in CUDA to run as super fast as possible. And the simulation runs basically on one graphics card at the speed of this movie. A few years ago, when we were using other platforms, it would take days to run these simulations. And it includes gene regulatory networks on the cells. It includes cellular behaviors, sort of anisotropic orientation of cell behaviors, also diffusion. Some of you may know simulating diffusion is actually one of the costly calculations because it's such a spatial one. And we aim to build, a complete, satisfactory, whatever that means, model of limb development, which has not been achieved yet for any organ. But what I'm going to talk about for the rest of the talk is one particular thing, and it's really about the very first thing. So it's just one side, it's the chemical side, as it were, the genetic side, and it's just one part of the pipeline. It's getting data. But it seems quite exciting to us. And maybe I should have called the talk, in fact, creating a 3D image without taking a picture. And hopefully you'll understand what I mean by that by the end. Because it is really about recreating something in space, but without directly imaging it, doing it in a slightly indirect way. So the world of spatial transcriptomics, of course, is massive and growing, and I think super important. Already 5 years ago, it was recognized as Method of the Year by Nature Methods. And essentially, there are sort of 3 or 2 main routes for doing this. There's this route here on 2D sections where you have spotted a whole load of barcodes, specific sequences in specific places, which then get incorporated into the library preparation from the RNA into the cDNA library. And indeed, this is a bit like creating a 2D image without taking a picture, right? Because you basically, once you've done this process, you can pull together all your cDNAs, and the information about where each one came from is encoded in the barcode. So you're not really taking a picture. This is maybe more easily seen by contrasting it to some of the other techniques which are image-based, because you're actually taking an image of the section while you're doing something, some chemical reaction, doing a sequencing reaction or an in situ hybridization reaction. So let's look at this one in 2D in a little bit more detail. And 2 years ago, in fact, there's a nice example that's relevant for us because it was a limb bud, in this case a human limb bud. So this paper does 2D transcriptomics on a limb bud, and although the shape looks a bit sort of hard to recognize from the previous slides, maybe you recognize that that is a limb bud. And here are 4 genes, and of course the technique has spatial information on thousands of genes. To go again quickly how it works, because it's the best way to introduce our new technique, it's on sections, and each sort of spatial region that you could think of as a pixel has been spotted with a unique barcode. So there's one barcode there, another one there, another one there. These are position-specific barcodes. It is known which sequence was spotted in each position, so that you can then afterwards, once you've incorporated these barcodes, pool all the cDNAs and just sequence the whole thing. So it's, that's very, very clever and very convenient. But we want 3D transcriptomics. 2D is not enough to understand most things. Of course, you can build up 3D by doing lots of 2D, but we did decide to explore whether there's a way of directly doing 3D. And instead of a sort of a 2D array of positions, we need to somehow specify a 3D array of positions. But this means specifying a 3D array of positions in the tissue, in an intact tissue. Now, the way the 2D thing works is because you cut the tissue into sections, and then each section can sort of have access directly to this array of barcodes. But we can't put a 3D lattice of barcodes into an intact piece of tissue. So, how are we going to do it? The idea was to do it somehow optically. And the idea was that if we could have, for example, 3 different fluorescent channels, and each channel had a sort of monotonic gradient of intensities, each in a different dimension, so one in X, one in Y, and one in Z. If these were really independent measurements that we could make, maybe we could somehow get this information into the cells while it's still in the tissue, and then, just as here you sort of pool all the cDNAs, if we could do this, we could actually dissociate the cells and pool all the cells. Because now it wouldn't matter to sort of keep track of physically where each cell had come from, because each cell would have a sort of a triple optical barcode. It's not a barcode, but an optical signal that would say where it had come from. Then instead of just putting it all through a sequencing machine, you would put it first through something like a fax machine, and then through a sequencing machine, or ideally sort of one machine that would do both. The first step would be, for each cell you could read where it had come from, and in the sequencing step you could sequence its transcriptome. So how are we going to do that? The idea was, well, we tried lots of different things, and this project has been over many, many years. But what basically has worked, and what was roughly the original idea, we want to label the tissue with strong fluorescent dyes uniformly. Uniformly is not what we want, but we need good, strong dyes, fluorescent dyes. And then to create these gradients in the 3 spatial axes, we would bleach them. So we'll first label the tissue, and then we'll sort of delabel the tissue, but in a controlled way. So that, for example, for one channel, you've got a monotonic, possibly even linear gradient in the X dimension. In another fluorescent channel, you have the Y dimension. In the third fluorescent channel, the Z dimension. What I'm showing here is not real data. This is sort of like a simulation, just a sort of an illustration. But this is what it would maybe look like for a limb bud. And you would also need, we realized very, very early on, you would need a 4th fluorescent dye as well that you do not bleach, because for a sort of internal control of the quantitative levels. Because basically 2 cells next to each other, if we're gonna infer what their position in space is based on very subtle differences in fluorescence intensity, but if one cell here and one cell here, one of them absorbed 30% less dye in general than the neighboring cell, then that would make it seem like it was far apart. So we need an internal control dye that will not be bleached, which will control for variations from cell to cell. And this basically is the result. So here we've got one limb. It's a particular structure. It's about 1 millimeter in size. You've seen pictures of them. And if we just first of all test what we call C3PO, and C3PO is the acronym for cellular 3D positioning by optical encoding, this is not doing the omics part. This is just to see whether C3PO works. And C3PO is the method of, in a sense, creating an image without taking a picture. So we dissociate the cells from the limb. If you put it through a fax machine, essentially every time a cell goes through, it's reading these 4 intensities. And 3 of these intensities are some kind of estimation of where that cell was in space. So, you know, from one channel, it tells you where it is in one dimension, from the other channel, another dimension, etc. And so the readout of these fluorescent intensities is some kind of estimation of where that cell was in 3D space. So you just do that again and again and again. Next cell, you read the values, its position is there. Next cell, you read the values, its position is there. Do it for lots and lots of cells, do it for as many cells as you like. The question, of course, and in fact the difficulty why it took years, is because there's so many reasons why this can go wrong. I mean, getting this gradients right is not easy. Dissociating the cells in such a way that they actually preserve the fluorescence intensities, but quantitatively enough that you haven't just lost all the information. Reading it out properly, correcting, etc., etc. But this sort of proof of the pudding is this. So if we just put lots of cells of this limb bud through and plot their positions in space, it looks like this. Now, it's not, you know, a perfect image, but actually what I'll try to convince you in this next slide, it is basically a limb bud, and it looks like a limb bud. What we did to get around doing the omics first was just to distinguish between mesenchymal cells and ectodermal cells. And this also helps because this should have a particular convex, sort of concave shape. So if we look at this point cloud of predicted positions of the cells and slice through it, this is a ring that is the ectoderm. This is like the skin around the organ. And this is what the skin should look like. And so you can see roughly the error, the spatial error of positioning of cells. But you can also see that it's not that we've just got a random ball of cells that are anywhere. We have an estimation of what this organ looked like without taking a picture. So we've repeated it multiple times. You can kind of reconstruct the shape of the limb bud, again, without without actually taking an image of the limb bud. And then what about the transcriptomics part? Well, of course, taking an image just for the sake of taking an image without taking an image, there's not much point. The point of doing that is so that we can then do things like transcriptomics. So in that case, you save each cell, and for each cell, you then, in principle, would have the transcriptome of the cell and its 3D location in space. The proof of concept that we've achieved so far is not cell by cell, but it's kind of binning spatially where cells came from. So these blocks that you see here, these are not physical blocks. We did not physically cut up the limb. It's just that when we read each cell, we said, oh, these cells are close to each other, so we'll pool these cells so that we can do the transcriptome on a few cells rather than just one by one. And this is sort of the result compared to what is the good old-fashioned test, the proof of what these gene patterns look like. So here you can see a bunch of genes. For example, this one, sonic hedgehog. It's in the posterior region, and C3PO predicts it to look like this. Here's BMP2, looks like this. IRX3 looks like this. And basically, I mean, we were completely astounded. This is not the resolution that one might want, ultimately, but astounded that this basically works. I mean, you can take a whole chunk of tissue, you can do this bleaching, dissociate it, and cell by cell try to work out retrospectively where it came from, and then you have the option to do transcriptomics and maybe many other omics on this data. So that is essentially how far we've got. We're trying to develop the technology further for things like developmental biology or the work that we do. This spatial resolution is actually extremely useful because all of a sudden we have access to the expression patterns at some resolution, at some spatial resolution, of the entire genome, and actually pretty cheap, given what the alternative would have been. So that's C3PO. You can see how it fits into our pipeline of what we're trying to do. It's just about getting good data. And this was almost all done by James Cottrell in the lab, also Jim Swoger and Alex Robert. So I want to thank them for this wonderful project, and thank you for your attention.
Okay, thank you very much, James, for the nice talk. So, is there any question in the room? We have time, because you stick on time. Yeah.
Hi, thank you for your presentation. It was very interesting. More curious than anything, where did the idea of the 3D positioning came from using the 3 gradients? Like, was it a random idea, or did you inspire from somewhere else or something?
Well, that's a very good point, and it was quite a long time ago. I mean, so the person that I listed on here, Jim Swoger, it was specifically him who came up with this idea. We had been thinking about alternatives. I mean, I think lots of people have been thinking about different ways to get this 3D data, and so in a sense there are variations. The idea that I had thought of does not work very well, which was using dyes but sort of putting them onto the tissue in different places and having then a kind of spatial gradient coming out from specific places. I mean, the idea that you want different fluorescent intensities and you want each space to have a unique combination of fluorescent intensities was sort of floating around. But Jim Swoger came up with the idea of bleaching. And bleaching is the clever thing. So we use a light sheet microscope, and you can literally calibrate it and bleach it in a very linear, both sort of linear directionally and linear intensity-wise, to make it a Cartesian coordinate system, so that it's, you know, you don't have to adapt it from tissue to tissue. It's just a general principle. Cartesian coordinates in 3D space.
Does it only work on very soft tissues, or does it also work on fibrous and bone and stuff like that? I could imagine it doesn't penetrate that well.
At the moment, it will not work probably on bone and fibrous tissue. But really, I hope this is in the early days, right? So we need to prove that it can work on some kinds of tissues, and then hopefully it can be improved. I mean, the limitations there, I guess, will be more to do with clearing, optical clearing of the tissue, and also dissociation techniques. I mean, this was also a non-trivial thing, how to, you know, you're fixing, labeling, bleaching, clearing, rehydrating, dehydrating, dissociating, and you also want all the RNA to still be sort of intact. I also didn't mention, but maybe it's obvious, maybe it's not. This kind of binning approach that I showed here is what we've achieved so far. Of course, the ultimate thing, which we think must be possible, is to do it literally cell by cell, like transcriptome for each cell, rather than binning it in these sort of large voxels.
Yeah, I was also wondering if there is an impact on the transcript integrity when you do the bleaching?
Well, there could be. I mean, I guess for that, what we've done some comparisons of the comparison for us would be to just take a limb bud and just do normal single-cell transcriptomics and compare it. I'm sure that it's maybe not quite as beautiful and perfect, but for us, the real sort of criterion of success is spatial patterns. So it's not necessarily, you know, the accuracy of the quantitativeness of the number of reads or things, although of course that contributes to it. But the problem is spatial. Right? And I think this is another thing that's interesting to talk about. When we get into spatial transcriptomics, you can kind of use spatial transcriptomics and then not use really the spatial element of it and still just go back to treating it like a whole bunch of cells. The value of space is that spatial organization is a higher level of of a biological complexity. That's what we're after, and that's what does seem to be captured here.
Okay.
This is maybe a little bit of a strange question, but could you swap one of the spatial dimensions for time, and would it make sense?
I mean, this is each sample is fixed and dead. Okay. I mean, for a start, it depends on dissociating the tissue to individual cells. So there's many reasons why we can't do it on living tissue. So we have to, you know, what I think is being done at the moment to go from 2D to 3D is what we would do to go from 3D to 4D. You know, at the moment you do many 2D sections to make 3D. Now we'll do many 3D ones to make 4D.
Any other question?
Thank you. A nice talk and interesting state-of-art also technology that you developed. I have a question regarding maybe the type of omics that you choose, because there are many layers of omics. Why specifically did you choose transcriptomics? Or do you think that adding other more layers of omics, like proteomics, could improve or develop our understanding of this biological mechanism?
I mean, that's a good question. So we're not fully aware of what the limitations for other omics would be, and we think that, indeed, it should be compatible with many kinds of omics. I mean, I guess it was natural for us to do transcriptomics because we just do a lot of transcriptomics anyway, and a lot of developmental biology is based on looking at gene expression patterns. So for us, it was the most natural one to go for. But, you know, ideas about, for example, clonal analysis based on even some genome sequencing or these lineage-based methods, in principle, if the cell is in a happy enough state, you could do any single-cell omics afterwards.
Any other question? Well, I have one curiosity. I mean, this is amazing how you developed this technique and everything, but my question is about which is the ultimate goal of having this. Once you have this technique properly designed, how this is related to your work with the lean development, which is the goal?
Okay. Let me show you. I mean, that's basically, if it will come up. I mean, it's basically this, right? So our goal is to make a computational model of this process, and it has to be based on reality, on what genes do over space and time. And so far, we and the field have been doing it kind of gene by gene, like getting to know which genes are our favorite genes and doing lots and lots of in situ hybridizations. Of course, we're probably missing a lot of the genes that are important. So if you can get to spatial transcriptomics, you can just get much more confident, let's say, coverage of the gene expression information that's relevant for all of this. But the goal is all of this, is building this computer model.
Okay, thank you. So thank you very much. Thank you, and the speaker.